What's up, guys, and welcome to another episode of the Food is Medicine podcast, in which we're going to be talking about atherosclerosis. Now, atherosclerosis is one of the leading causes of death in many developed countries, especially the United States. And atherosclerosis can be characterized as a plaque buildup in the arterial walls that can lead to some heart diseases like coronary artery disease, and this plaque can restrict blood flow. Now, to talk about atherosclerosis, we really need to dive into cholesterol, lipoproteins, and pretty much the bad guys that kind of cause atherosclerosis. All right, so let's really dive into what cholesterol is. So cholesterol is part of the sterol family, and the sterol family is characterized as the 4-carbon ring structure. Now, cholesterol is one of, if not the most important lipid macromolecule in human physiology, because cholesterol is responsible for plasma membrane fluidity. That being said, the plasma membrane is a lipid bilayer, so it really needs to stay fluid in order for the cell to basically carry out many processes like cell-to-cell recognition, signal transduction. So there's many important things that the cell needs to perform in terms of function that cholesterol helps with in terms of the plasma membrane. Now, second off, cholesterol is a precursor to many hormones such as testosterone. So precursor, and let's do two hormones. Now, as we all know, testosterone and estrogen are very important for human physiology, and cholesterol is a precursor of those hormones. All right, so now we got cholesterol down. So now let's talk about lipoproteins. So when we're talking about lipoproteins, we're talking about LDL, or the low-density lipoprotein, and we're also talking about HDL, or high-density lipoprotein. Now, the low-density lipoprotein is basically saying that it has a low density of proteins. So low-density lipoproteins are usually going to be less dense and bigger in size, while our high-density lipoproteins are going to be in have a higher concentration of proteins, and they will be smaller but much more dense. Now, it's crucial to keep in mind that LDL and HDL both contain cholesterol. Now, LDL has more cholesterol, and in lipid panels, it will be denoted as LDL-C, and for HDL, HDL-C. So in usual lipid panels, you'll get your total cholesterol, triglycerides, and LDL cholesterol, and HDL cholesterol. Now, like I said, LDL usually has more cholesterol, and in these lipid panels, if you have a higher level of LDL cholesterol, you are at more risk for cardiovascular events. And there's this preconceived notion that LDL cholesterol is the bad cholesterol, and HDL cholesterol is actually the good cholesterol. Now, a lot of people mix up LDL and HDL from LDL-C and HDL-C, that meaning cholesterol. So we really have to separate that LDL, low-density lipoprotein, is a lipoprotein. That is the thing that transports cholesterol. So you can think of cholesterol as cargo, and the LDL, or the lipoprotein, is the ship. So the ship carries the cargo around the entire body, wherever the body needs cholesterol. Same thing with HDL. So that lipoprotein is our ship, and the cholesterol is the cargo. So we really have to keep that in mind. And when we're looking at LDL and HDL and saying which one's bad or good, this is pretty much an incomplete description, because LDL, the lipoprotein itself, is what could be atherogenic, and we'll talk about what can make LDL atherogenic. Atherogenic meaning that it can cause atherosclerosis, and HDL, the lipoprotein itself, is a good lipoprotein because it, in a physiological standpoint, it acts as pulling cholesterol out from arterial walls, and it has a higher density of proteins. So This whole notion that LDL cholesterol and HDL cholesterol, that LDL cholesterol is bad, HDL cholesterol is good, is an incomplete description, because we're talking about the lipoprotein that's either good or bad, not the cargo inside. We're talking about the ship, because the ship is responsible for delivering the cargo. It's not the cargo's fault of if it's bad or good, but rather the ship and how it acts. So now we know that. Now we know LDL cholesterol, HDL cholesterol, and there are other lipoproteins like VLDL, IDL, and chylomicrons, but we'll talk about those in a later podcast episode. But today I really wanted to elucidate LDL and HDL. So just remember, LDL cholesterol, HDL cholesterol, there is no such thing as good or bad. It is a lipoprotein itself that we need to be concerned about. Now, with LDL cholesterol, in most LDL cholesterols, most LDL lipoproteins, there's going to be this protein known as Apo-B. And the Apo-B is a protein that's attached to lipoproteins, to these LDLs. So if I could draw this as a rough sketch, and please be patient with my drawing because I'm not a artist in any way, but you could think of let me put some cholesterol in here. So let's just say these dots are cholesterol, and over here, this is our LDL, and our Apo-B is this protein on our LDL. So Apo-B, or apolipoprotein, is very important in that it can be atherogenic. So Apo-B can also be on VLDL, IDL, and chylomicrons, but it is mostly on those LDL lipoproteins. That's what can make an LDL lipoprotein atherogenic, when it contains this Apo-B protein. And like I said, atherogenic meaning that it can cause atherosclerosis. So one of the biggest risk factors of atherosclerosis is having a high particle number of LDL. So number of LDL, if your LDL particle numbers are high, then that could put you at higher risk for atherosclerosis. And here's why. Because when we have these LDLs, we will usually have the Apo-B protein on that LDL, which can make the LDL atherogenic. And if we have a lot of these low-density lipoproteins that have that Apo-B protein, then this can pretty much increase our chances of having atherosclerosis, because these LDLs will be circulating around the blood, and this pretty much is, you could think of it as having more bad guys, because it is the LDL with that Apo-B protein, and that's what can cause atherosclerosis. Having a high number of LDL particles increases the risk of atherosclerosis, because you have so many bad guys around here, and this red line is pretty much replicating our arterial wall. And these bad guys, when there's so many of them circulating around the blood, there's a higher chance for crime to occur. And these LDLs can actually go into the arterial wall. And let me try to draw an LDL inside the arterial wall. And here, let me just put some dots around there. Let me put our Apo-B protein around. So the LDL can go inside the arterial wall. And there's actually one thing that occurs, is that there doesn't need to be inflammation or injury to occur. So these LDLs with the Apo-B protein, they can actually transcytose into the arterial wall. So there doesn't need to be any inflammation or injury to the arterial wall. But when these LDLs, with those Apo-B LDLs, get into the arterial wall, they can increase inflammation. Now, you may be wondering, well, how do these LDLs actually increase inflammation? Because we know that they can go by transcytosis. So let me actually write that real quick. And transcytosis is a way that these Apo-Bs can go inside the arterial wall. So the question is, how does inflammation occur in the arterial wall? Well, we have our immune system, and we have these immune cells known as macrophages. So let me just draw that. Macrophages. So the biggest purpose of macrophages is to phagocytize, or basically consume and engulf anything that may be deemed as a pathogen or something that's not supposed to be there. So these macrophages will come across that LDL and try to consume it or engulf that LDL, because that LDL, that Apo-B, is not supposed to be in the arterial wall. Now, unfortunately, when the macrophage does try to attack these LDLs, and actually let me put a couple more LDLs. So the macrophage is going to try to attack these LDLs, and there will be immune response that occurs, and foam cells can be developed. So biggest thing to realize in many diseases is that these diseases are a consequence of our immune system trying to attack a pathogen. For example, pneumonia. The reason why we see a lot of fluid buildup in the alveoli, or those air sacs in the lungs, is because of the immune system trying to fight off, I believe it's pneumococcus, or it's, I forgot what bacteria it is, but it is a bacteria that the immune system is trying to fight off. And the consequence of that is that there's a fluid buildup in the lungs, as well as some other symptoms. So when these macrophages try to fight off these LDLs, there will be an immune response that occurs, which increases inflammation, because as we know, the inflammation is further stimulated when there is a quote-unquote war going on with the immune cells. So inflammation will occur, and things like interleukin 6 or these cytokines will be released to bring more macrophages in. What happens is foam cells will develop, and these foam cells are just these macrophages that have consumed these lipoproteins, and this is what can result in that plaque. So there's this model known as the response to retention model. Now this basically shows the response to retention model, and it is that for the first thing that needs to occur, let me change my color over here. So for the first thing that needs to occur for an LDL to cause atherosclerosis is for it to get in, for these Apo-Bs to get in. So that's the first thing that needs to occur. The second thing that needs to occur is that these LDLs need to be retained. So these LDLs, these Apo-Bs, they need to be retained in the arterial wall. And then the third thing that needs to occur is this inflammatory response, or this oxidation of LDL, oxidized LDL, and this immune response needs to occur. So this response to retention model is basically telling us that this LDL needs to go into the arterial wall first, then it needs to be retained, then it needs to initiate this immune response to create these foam cells, and that's when atherosclerosis will occur. So it's this three-step process that needs to occur for atherosclerosis to happen, or more specifically, for this plaque to build up. Now, what I've seen around the mass media is this push of small LDL being worse than large LDL. And if we actually do look at the case studies, it is true. But they don't really compare the large LDL and the small LDL together. And this is when the terms of concordance, and discordance come around. So to put this in layman's terms, concordance is when two factors agree with each other, and discordance is when two factors disagree with each other. So an example of this would be LDL-C and the number of LDL particles. So let me actually write that real quick. So LDL-C, that meaning LDL cholesterol, and LDL, that meaning the low density lipoprotein. So in concordance, according to concordance, we'd see that if there's an increase in LDL cholesterol, we would usually see an increase in the number of LDL particles. And that's concordance because it's showing us that those two factors agree with each other. Now, discordance, we could see an LDL cholesterol that is low, and then the number of LDL particles being high. So then the question comes, which one should we actually prioritize? The LDL cholesterol or the number of LDL particles, which we've ruled out that it is the LDL particles that really matters, because the ships that go around the bloodstream are more important than what's actually in the cargo. Because without the ships, the cargo can't go anywhere. Cargo being the cholesterol. Okay, so going back to these small LDLs and large LDLs, the reason why we've seen that small LDLs corresponds with more cardiovascular risk is because, one, we've seen these in patients with diabetes or metabolic disorders, in which you will see a lot more of these small LDL particles than compared to large LDL particles. And there's more of this suppressor effect, in which when we're not really comparing the large LDL and the small LDL side by side, we're not really keeping the particle size in control. Because sure, we could say that small LDL is worse than large LDL, but usually when we say this, there's more small LDL particles than large LDL, because you could have, like, for example, 1, 2, 3, 4, 5 small LDL particles, and then 1, 2, 3 large LDL particles. So there's this suppressor effect in which we're not really comparing large LDL versus small LDL, and we're only seeing small LDL, and we're basically saying that because these LDLs are small, they can go into the arterial wall easily, and this can lead to atherosclerosis, which if we look at, because we've been looking at that through diabetes and metabolic disorders, but if we look at that through familiar hypercholesterolemia, or FH, in which it's a genetic disorder in which there are large amounts of these LDL particles, we're basically controlling for that LDL particle factor. And when we're comparing small LDL and large LDL, there's nothing intrinsically wrong with a LDL being small versus an LDL being large. And it is the particle size that really matters. Hopefully that made sense to you guys, but if it didn't, I'll link a video down below talking about small LDL versus large LDL. But just to sort of summarize, size is still important, but it is an incomplete description, because the number of cargo ships actually matters. Now, those cargo ships can be smaller, and maybe it can enter places a little bit more, but those are like having smaller bad guys. You could have large bad guys and smaller bad guys, but if you have the same amount of bad guys, they can still do the same amount of damage. And like we said, the injury and inflammation doesn't have to occur for these LDLs to enter the arterial wall, because they can enter through transcytosis. So that's pretty much looking at small LDL, large LDL, and atherosclerosis is, by plain definition, it's when a sterol enters an arterial wall macrophage. So what this basically means is, if we go back to our diagram and we look at the response to retention model, the Apo-Bs, those LDLs, have to enter the arterial wall first. They have to be retained in there, and then they have to oxidize, or they have to go through that immune system response, get consumed by that macrophage to form those foam cells, and then a plaque will eventually form. Now, this doesn't mean we have to throw out LDL-C or particle size as a factor for cardiovascular risk, but it does show us that the number of particles actually matters a little bit more than LDL-C and our particle size. Hopefully that made sense, and there will be more videos on atherosclerosis and talking about other lipoproteins like VLDL, IDL, chylomicrons, and Lp(a), which is a really cool one. But that concludes our video for today. Hopefully you guys learned something, and as always, a Pomodoro a day will make you a doctor someday. I'll see you guys in the next one.